Good morning and welcome to the Helio Genomics virtual KOL event. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player. As a reminder, this call is being recorded and a replay will be made available on the Helio website following the conclusion of the event. I'd now like to turn the call over to Justin Lee, Chief Executive Officer of Helio Genomics. Please go ahead, Justin.
Thank you, Tara. Good morning, everyone, and thank you for joining us. We're extremely excited to share significant advancements in the field of liver cancer diagnostics and precision oncology as Helio Genomics has continued to make breakthroughs in both validating our flagship HelioLiver DX test and pushing towards our robust pipeline of products. Our vision is to become the leading provider of cfDNA blood tests for the entire cancer journey, from screening to treatment guidance to recurrence monitoring. We utilize advanced multi-omic signals found in blood that are indicative of tumor biology. Our passionate and dedicated team has already made early-stage HCC detection a reality, with the help of thousands of patients and hundreds of physicians across the nation. We look forward to expanding to new horizons, and we hope you'll enjoy our presentation today. By way of introductions, my name is Justin Chen Lee, and I'm the CEO of Helio. After getting my finance degree and later my MBA from UC Berkeley, I worked for various Fortune 500 companies. In 2017, I joined Helio as one of the founding team members and helped spin out our foundational intellectual property from UC San Diego. After wearing multiple titles throughout the years, including VP and GM, I took over as CEO in 2021. I also serve on the leadership board for the UCLA Terasaki Institute for Biomedical Innovation and as a venture partner for the SC Master Fund. I'll pass it over to Rick to introduce himself.
Good morning. I'm Rick Van Etten. I'm serving as Chief Medical Advisor for Helio Genomics. I'm a hematologist oncologist, MD PhD from Stanford. Spent the earlier part of my career at Harvard Medical School, Dana-Farber, and then at Tufts. About 11 years ago, I came to the University of California, Irvine, out here in Orange County, to become the director of the National Cancer Institute designated Comprehensive Cancer Center at the university. In that role, I have global responsibility for cancer research across the entire university. I've been working with Helio since about 2018, and as Chief Medical Advisor, I play a role in helping the company develop its technology, particularly translate its technology into tests, including design of clinical trials for validation.
Gary?
Hi, Gary Frazier, Chief Growth Officer for Helio. I have been part of the company since January of this year to kick off the commercialization strategy. My background is I'm a recovering hospital administrator. I work for several health systems, from Dignity Health to Presence Health, which is now part of Ascension in Chicago, and many more. My role typically was always strategy. So I was Chief Strategy Officer, VP of Business Development, responsible for physician alignment, growth, strategy around service lines, as well as having my own startups a few times over the course of that. Thank you.
Shivani?
Hi, everyone. I'm Shivani Mahajan, the head of the computational science team at Helio Genomics. I've been with Helio for about 3 years now. Just a quick word about my background. I have a bachelor's and master's in engineering in bioinformatics and mathematical modeling, following which I did a PhD in UC Berkeley in integrative biology with a focus on functional and evolutionary genomics and computational biology. Following that, I did a postdoc at UC Berkeley and then made my entry into industry by working at Freenome. So Freenome was my first industry position and my foray into the liquid biopsy space. And I stayed there for about 3.5 years, working on various analytes and trying to build machine learning models for colon cancer detection, and joined Helio carrying that knowledge forward and working on liver cancer.
Go ahead, Dave.
Hello, everyone. I'm Dr. David Taggart. I'm the Chief Scientific Officer here at Helio Genomics. I've been within the company approximately 10 years. I have a PhD in biochemistry from the University of Wisconsin in Madison. I also hold a certification in clinical chemistry. A quick word about my background. I published manuscripts on a variety of topics, including manuscripts describing virus gene expression and genome replication, manuscripts describing the underlying mechanism of how DNA polymerases generate errors that leads to specific mutations that drive cancer and oncogenesis, manuscripts that describe the development of novel drugs to treat liver cancers, and of course, manuscripts that describe the development of novel diagnostic tests to detect cancer. I also am the author of patents or patent applications in these same areas. Here at Helio Genomics, I work to drive the overall research strategy and coordinate activities cross-functionally among our various departments and also with external shareholders. Alex, do you want to go next?
Yep. Thanks, Dave. Yeah, great to meet everybody. My name is Alex Brenner. I'm Chief Financial Officer. I've been with Helio now for about 5 years. Prior to joining, I spent the bulk of my career working for Fortune 500 companies, running operational finance for $100 million plus business units. Spent quite a bit of time doing M&A activity, strategic market planning, and infrastructure development. I took a break from corporate America, spent 2 years running finance for an ed tech nonprofit in Seattle, where I'm based. Working in there, I got a lot of energy out of working for a mission-driven organization. And so when it was time to rejoin the for-profit world, I was looking for something that could fill that box for me. So the chance to join Helio and help solve cancer was right where I wanted to be. So happy to be here. And with that, I'll pass it back to Rick.
Thanks very much, Alex, and good morning again, everyone. I'm excited to take a few minutes to just walk you through the HelioLiver DX test and what it's all about. So Helio Genomics has a series of platform technologies that have been essential to developing this test and other future products. They're shown here. All of them have been published in peer-reviewed journals. The Eclipse is Helio's wet lab platform, which is the proprietary method for actually analyzing the methylation signals in these trace amounts of circulating tumor DNA that can be found in the blood. The proprietary technology uses a different method, different than bisulfite. It's an enzymatic method to convert the methylcytosines into a signal that can be detected by next-generation sequencing, and that allows greater sensitivity for the detection of these elements in the blood. MESA is shorthand for multimodal epigenetic sequencing assay, and it recognizes that additional factors in addition to the methylation signature can add value to the predictive information found in cell-free tumor DNA. Specifically, the approach combines what's called nucleosome occupancy and DNA fragmentation into the algorithm. And then lastly, Chao, which is adjusted clonal methylation, is a technology developed by my UCI colleague, who's also a Helio consultant, Dr. Wei Li. And what Chao does is basically summarize the epigenetic information and is able to distinguish what we call cis from trans, which is where two methylation markers are on the same strand of DNA versus being on different strands of DNA. And that also adds significant value to the power of the algorithms. So there's a huge unmet clinical need in hepatocellular carcinoma. Hepatocellular carcinoma, HCC for short, is one of the few cancers that's actually rising in incidence and mortality in the United States. There is a high-risk population. It is people that have liver cirrhosis or chronic hepatitis B, with or without cirrhosis. They have an incredibly high rate of going on to develop liver cancer. Annually, between 1% and 8% of this population will develop a liver cancer. So the current guidelines from advisory bodies such as the American Association for the Study of Liver Disease is that these patients should undergo surveillance. And the current standard is to have ultrasound, abdominal ultrasound, with or without determination of alpha-fetoprotein, which is a circulating blood marker. And this should be done every 6 months. And numerous studies have shown that early detection improves patient outcomes. This includes progression-free and overall survival. The problem is that the performance of ultrasound is not optimal, and there are a couple of reasons for this. The top reason may be that patients with cirrhosis don't get ultrasound at the recommended frequency. The real-world adherence rate varies between 20 and as low as 9%. There's a couple of reasons for that. Sometimes there's access for testing is limited, particularly in rural health locations. Furthermore, ultrasound has low sensitivity for small liver lesions, which are the most important to detect because they're the most treatable. And lastly, the performance of ultrasound is influenced by the operator expertise and also negatively impacted by obesity, which is a growing problem in the United States. So it's clear that additional methods for hepatocellular carcinoma detection are really needed to overcome these weaknesses. That is where the HelioLiver approach comes in. The process starts with phlebotomy, with a simple venous blood collection, where we collect both serum and plasma in a kit. This is collected. There's some routine processing that takes place to separate serum, and then these analytes are then basically stable and can be shipped at room temperature. At the lab, the analysis is broken down into two parts. One part is to quantify the levels of 3 protein biomarkers. So that's alpha-fetoprotein or AFP, the L3 fraction of AFP, and DCP, which is carboxyprothrombin. This is done by immunoassay by standard methods. The other part of the assay looks at the circulating free DNA methylation based upon the platforms that I talked to you earlier. The two are then combined together in a diagnostic algorithm. And the results internally are actually quantitative. There's a number that comes out. But in terms of reporting to the test, the person that orders the test, the reporting is qualitative. The results of the test are reported as either positive or negative. Having developed the test, the company then conducted a case control study of its performance in a group of 247 subjects, about half of whom had pathologically confirmed hepatocellular carcinoma and about half as controls. And you can see that at a specificity that's very high, 91%, the HelioLiver test demonstrated higher sensitivity than any of the other commonly used blood tests, such as AFP, or GALAD, which is actually a combination of those 3 markers. And this was true both for all stages, where the sensitivity was 85%, and particularly for early stage, AJCC stage 1 and 2, where it was 76%. So this was very promising, but it doesn't really assess the performance of HelioLiver, or indeed any diagnostic test, in a real world setting where patients are assessed prospectively. To address that, Helio Genomics launched the CLIMB study, which is the first of its kind prospective multi-center blinded trial of comparing the HelioLiver test with ultrasound and AFP. So the schema is shown on the right. Subjects were enrolled that either had cirrhosis or chronic hepatitis B, and all subjects at registration had all 3 tests, HelioLiver, ultrasound, and AFP. All subjects then went on to have multiphasic MRI with contrast, which is the way to determine in this trial the clinical truth. MRI results are actually binned according to the LIRADS protocol, and there are several different categories as shown here. For the purposes of the trial, LIRADS 1 or 2 were called negative, whereas LIRADS 4 or 5, or cases that showed tumor and vein characteristics, were called positive. That leaves a category called LIRADS 3, which is indeterminate. And in order to adjudicate those subjects, everyone went on to have follow-up MRIs at frequent intervals. And they were, again, binned as either positive or negative. These are just the demographics of the subjects in the trial. There was a male predominance. There was a fair proportion of people with Hispanic ethnicity and obesity and diabetes. The study was carried out at both community-based sites and academic sites in a ratio of about 4 to 1. And there was a broad range of etiology of the underlying liver disease, most frequently non-alcoholic or metabolic associated fatty liver disease, but also alcoholic liver disease, and some cases of viral disease. This is just the disposition of the subjects who were enrolled. Initially, 1,968 participants were consented. Of those, the initial 412 that were enrolled prior to June 20th, 2020, were assigned to a training cohort. The remaining 1,556 went into the trial per se. Of those, 271 were not evaluable, mainly because they did not undergo either ultrasound or MRI at their initial visit. That left 1,285 in the main trial. In this population, 46 hepatocellular carcinomas were diagnosed, 42 at the initial assessment, and 4 in the follow-up of patients that had the LIRAD-3 indeterminate characteristics. That's a frequency of about 3.6%, which is entirely within the range of what was expected. These are just the characteristics of the tumors that were detected. You can see that the majority had only one solitary lesion, and about 46% had small lesions, which is defined as less than 2 centimeters. So HelioLiver DX met both its primary and secondary endpoints. The primary endpoint was to show at least a 5% increase in sensitivity over ultrasound and to be non-inferior, where the non-inferior margin was predetermined to be minus 10%. These are just the point estimates on the left for sensitivity, on the right for specificity, with the 95% confidence intervals. So the primary endpoint was for all lesions. The secondary endpoint was for lesions that were less than 4 centimeters. And there was, in addition, an exploratory endpoint for the smallest lesions of less than 2 centimeters. And you can see for that group, it was almost 29% sensitivity. If you break down these results based upon tumor size, you can see that on the left, you can see comparing to ultrasound alone, the HelioLiver test was both superior and non-inferior, as I mentioned, to ultrasound alone. It was also, not sure if this slide is animated or not. No, it's not, sorry. It was also superior to alpha-fetoprotein alone. If you break these down by lesion size, you can see that the HelioLiver test outperformed ultrasound when it mattered the most. And by that, I mean in detection of early lesions. So you can see that for T1 lesions, according to AJCC, HelioLiver vastly outperformed ultrasound. And then on the right-hand side, if you look by largest lesion size, for lesions that were between 2 and 3 centimeters, it was significantly better. And importantly, for the smallest lesions, the sensitivity was about 30%. You can see here that ultrasound failed to detect any of these early lesions, which I think is critically important. This is just a summary of the overall test characteristics. And you can see the overall sensitivity of ultrasound in the CLIMB study was 28%. For those of you who know, if you look in the literature, there's a very wide range of sensitivities reported for ultrasound in this setting. It can range from 20% upwards of 80%. Many of those studies are actually case control studies, which are biased by the pre-selection of patients that already have liver disease, many of which were actually detected by ultrasound. And also some of them reflect studies where the sensitivity of ultrasound is actually a cumulative sensitivity with multiple tests. This level of sensitivity is entirely concurrent with a Korean study, the PRIUS study, which is the other prospective study of ultrasound performance and surveillance, which arrived at a virtually identical number for sensitivity. So HelioLiver addresses several key ultrasound weaknesses. The issue of accessibility to screening or surveillance is addressed because this can be applied with any clinic blood draw, whereas, as I mentioned, ultrasound requires the availability of specialized equipment. It's independent of the skill and training of any operator. It can detect very small lesions, and most importantly, obesity or elevated BMI has no effect on the performance of the test, where it greatly denigrates the performance of ultrasound. So in conclusion, the CLIMB trial met its co-primary endpoints, was more sensitive than ultrasound for detection in this high-risk population in the very early HCC lesions, less than 2 centimeters. And we believe that this blood test has the potential to greatly improve patient outcomes, both through improved adherence and superior early detection of hepatocellular carcinoma, which are those lesions that are most amenable to curative therapies. We also believe that the test has a role beyond just diagnosis, which is to predict outcomes and prognosis in patients with liver cancer. And the same circulating free DNA methylation score as used in HelioLiver was assessed for its ability to predict response in early stage liver cancer. And this was in collaboration with our colleagues at the Ochsner Clinic in New Orleans in Louisiana. They had a cohort of 108 patients who had early stage liver disease that were aiming for intervention. And the interventions ranged from transplantation to resection to local regional therapies. And we just segregated those patients by their response and their methylation score. And you can see on the right-hand side, there was a pretty clear separation between those patients that were able to receive these therapies and be free of disease progression versus those that progressed. These data are exciting because they suggest that the methylation information that's captured in these circulating free DNA tests like HelioLiver actually has predictive power in addition to diagnostic power. The data were presented that are here were presented by Dr. Mahajan as the top-ranked abstract at the San Antonio Liver Cancer Conference in October this year. So the current test status, which Gary will go into more, is that it is currently available as a laboratory-developed test with a CPT code and is covered with prior authorization by most health plans. The CLIMB data itself has been submitted to the FDA, and as Gary will mention, we're expecting a decision in 2025. The current price of the test is $662.32 set by CMS. Reimbursement obviously depends on insurance. And Dr. Mindy Nguyen, who's the chair of our Hepatology Advisory Board from Stanford University, is currently conducting a cost-effectiveness study of HelioLiver versus ultrasound. And Gary will show some slides about the expected benefits. The pipeline that Helio Genomics has developed has broad applicability across cancer. The biomarker discovery using their automated platform to identify methylation patterns for different tumor types, and the proprietary algorithms used to train the test allows the technology to potentially be applied to many different types of cancers, both for diagnostic purposes and, as I mentioned, for treatment response predictions. And lastly, for MRD and disease monitoring. And we're already exploring those applications in liver cancer. And this is just the current pipeline focused on liver cancer. At the top is HelioLiver, which I already mentioned. We're expecting potential FDA approval in the third quarter of 2025. We are also continuing to refine the test based upon the data that we are actively collecting in real time. And that will form the basis of what we call HelioLiver 2.0. And the aim there is to improve its test performance characteristics. The precision oncology application is the one that I just showed you that is able to predict outcomes in early stage liver cancer. And then we have additional applications in development for monitoring disease response and minimal residual disease. So I'd like to thank you for your attention. I'll be available to take questions at the end, and I will now turn it over to Gary Frazier, who's going to talk about commercialization.
Thank you, Rick. So every growth strategy should start with the patient population that you are targeting or that you are working with. And in our case, it's no different. Our primary market, as you can see on your screen, which is upwards of 1.7 million liver cirrhosis patients and approximately 700,000 hepatitis B without cirrhosis patients, that's considered, as Rick mentioned before, the high-risk population, those that are under AASLD and EASL guidelines for every 6 months. What drives that market is also a consideration because there are many patients that are on the cusp, if you will, or have other demographics that add to whether they should or should not be surveillanced, and that is the non-alcoholic, NASH. And so when you look at these kind of markets, you say, well, okay, they're not as big as, say, colorectal, but size matters in so much as incidence rate. And so the incidence rate for a colorectal, as you all know, is sub 1%, whereas an incidence rate, as Rick mentioned earlier, for cirrhotic patients for HCC is between 1 and 8%. And so that's factored into it, as well as the frequency of testing. Being tested every 10 years for every person over 45 is different than being tested every 6 months for everyone with cirrhosis. And so all of these factors lead to a unique patient population, which means it has to have its own unique tactical approach to growth. This also leads us to say, okay, well, who's the payer mix? Or what's their payer mix is the first big question, so that we can develop a unique payer and contracting strategy around this. And so when you look at that very population, based on diagnosis codes, you see that a large majority of them are in the private category, and another large chunk is within the Medicaid category. And really smaller percentages are in that uninsured Medicare and other category. Tucked in the private are also Medicare Advantage, which again drives some of how we go to market, what we do, what payers that we partner with early on with the LDT versus what we do in the long future. And so therefore, when you think about, people say, well, MolDX, we will be going through that process. We're slated to go through that process in Q1, but it does not drive or hinder our growth strategy and go to market today with our LDT, because as you can see on the pie chart, we can go to market, and as you'll see in the next slide, we've made quite a bit of progress with our payer and contracting strategy. So here's where we stand today. And this is an iterative, ever-expanding process, as all of you know, especially those of you who are practicing day-to-day in healthcare with coverage. We have where the CPT code at the top of your screen with the Medicare rate of $662.32 is a covered benefit for many of the largest national payers, such as BCBS, Blue Cross Blue Shield, Anthem, Humana, Kaiser, as you see on your screen, many large MCOs like Centene that go by multiple names in different states that you know of, some with prior authorization, some without prior authorization. And then on the state side, which is the Medicaid population, we've made some pretty good progress there, as you can see on your screen, with California and Arizona and Michigan and Missouri and many others. And we're actively being credentialed for another 5 to 6 states as we speak, and soon will be a covered benefit in many of those states, such as Louisiana, Georgia, Indiana, and Illinois. And there are a few more that we're working on. And so this particular approach, again, allows us to go to market with our LDT, negotiate with the various plans, not just around authorization, prior authorization or non-prior authorization, but even around our rate. Starting with that Medicare rate, we can, in some cases, offer discounts, which puts us in the ballpark of an ultrasound, believe it or not. And so this is not a uber expensive next generation sequencing test that puts us way out of market or that patients can't afford. In many cases, there will be no out-of-pocket for a patient. In most cases, for the plans, this is a head-to-head matchup between our tests and ultrasounds, to where there isn't that much of an economic difference. And I'll go more into that in a future slide. So our approach to really the market at large, and again, to use the Cologuard as a contrast, is that we're not going direct to consumer. It doesn't make sense to go direct to consumer with HelioLiver. It did make sense to go direct to consumer for Cologuard for various reasons. Again, they're targeting the entire population over the age of 45. They had an uphill battle when you're going against colonoscopy, which is a a modality that the very physicians who would be administering the test also use or make money with, right? And when it comes to colonoscopy. In our situation, when we're working with cirrhotic patients and their physicians, which are mostly hepatologists, most of them are not doing on-site ultrasounds in their practices. Most of the time, they will write an order, and the patient has to leave the practice, schedule an appointment to get their ultrasound, which leads to the low adherence rate and the fallout or the fall through of those patients almost never going to get that ultrasound. In our situation, we are able to work with those physicians to improve adherence rates, work with those health plans to improve adherence rates, which ultimately helps all of them, the patients included out, which puts us into a space where we could work with managed Medicare organizations, managed Medicaid organizations, work with accountable care organizations. We like to target and work with IDNs, integrated delivery networks, because this gives us more of a B2B approach to marketing, to growth, and to sales, which leads to a more efficient go-to-market strategy because we don't need teams of hundreds of people, boots on the ground, going bottom up, clinic to clinic. We could work with organizations. They all get and understand our value proposition. They all get and care about the cirrhotic population or the high-risk population getting the care and not developing HCC unknown until it's the late stage, and there's very few things that they can do. And the therapies that are available at late stage, as you all know, are much, much higher cost than those at early stage. And so, when you look to your right, evidence is the flywheel. Working with these organizations, proofing through real-world utility that the test not just works from a efficacy perspective, but also an efficiency perspective, as in more people are able to have the test done during their regular visits because it's just a blood test, improves that adherence rate, which improves the likelihood of actually diagnosing HCC in the population. And so to just conclude on this slide, one of our clients, for example, covers 2 million lives. Out of those lives, they have 20,600 cirrhosis patients, so just about 1%. And if we move the adherence rate from where it is at 10% upwards to 50%, times the cost of our test, then we can actually calculate the value to this company of the actual agreement and partnership with that MCO at $13 million per year in revenue. But more importantly, we can improve the care within that organization. This is just a quick highlight on the economic conversation. On the left-hand side, we've touched on it before, the cost differential between early stage as well as late stage, because just the options, the therapeutic options there, from ablations to resections to even transplant, are all available at the early stage and therefore give you a much lower range of treatment cost versus late stage. But when we look at just the ROI, if you look to your right, partnering with the health plans, especially the Medicare Advantage plans that use what's called a RAF score, risk-adjusted scoring system, there's a positive ROI, primarily because for Medicare Advantage plans, every new diagnosis means more money to the plan from the federal government. So an HCC diagnosis, for example, is another $13,000 to the plan for that person per year. If you can catch the HCC or you can diagnose the HCC when they have years to live, it's more revenue versus diagnosing when there's 3 to 6 months to live. There's not more revenue, there's just higher cost. And so again, the economics, even on the long-term view for all of the patients, is a positive for most of the health plans that we're working with. And this is my last slide here, just to kind of give you a redacted view of our pipeline. Again, we're working with health systems, MCOs, integrated delivery networks, ACOs, and some ERISA employer plans. And we're doing this because at the health plan level, there's economic incentive for the physicians that they are either employing or tightly affiliated with, like a narrow network. There's a clinical value there. And then, of course, there's a patient impact. And so when you look at our pipeline, we've already closed and have onboarded our very first client as of last month. Into this month, we're actually implementing, and they have begun testing, and we will see those kits coming back to us sometime next week. And so this is fresh off the presses, and that's here in Q4. We're expecting another three health systems to roll through by the end of this year into early 2025, and it will continue to roll that way. These health systems are named health systems. They span from the Southeast to the Midwest, all the way to where I am now, which is Southern California and then Northern California. And so we've got a good swath of the United States represented in our pipeline. I will now pass it on to the Q&A session. Thank you very much.
Great. Thank you, Gary. So at this time, we'll be conducting a question and answer session with our speakers. As a reminder to our audience, if you'd like to submit a question, please use the Q&A text box at the bottom of the webcast player. Justin, I'll turn it back to you to moderate the session.
Thanks, Sarah. Okay, so it looks like we have our first question here. Can you talk a little bit more about the MESA technology and how and if AI is incorporated into that? Dr. Mahajan, would you like to take that?
Yes. As Rick mentioned, MESA stands for Multimodal Epigenetic Sequencing Assay. And so the idea here is that within a single assay, we can derive and study multiple kinds of epigenetic features of cell-free DNA. So because of the fact that during the wet lab part of the process, we use an enzymatic conversion step, this not only allows for higher yields and lower starting material, but it causes less degradation of the DNA. And so this makes it possible to now not just study cell-free DNA methylation from this data, but also to look at signals like fragmentomics and nucleosome occupancy. And using AI, we have found that when we try to extract and features of different kinds and then combine them in machine learning models, we find that the signal is stronger when we use multiple modalities, like I just mentioned, rather than just using a single modality alone. And we demonstrated this as a proof of concept study in colon cancer that was published, previously. But the AI component essentially involves the part where we are combining these signals together.
Thank you. Next question. What does it take to get to scale in terms of infrastructure in later years? I'll invite Alex Brenner, our CFO, to answer that question.
Yeah, I mean, we're really looking at multiple channels. From an internal perspective, we can scale up the lab from a hard CapEx standpoint from about $1.5 to $1.8 million to replicate our line. So we can add more to our existing footprint, and each line that we adds on average about $30 million of revenue on an annual basis. Similarly, we're looking at potential partnerships that could help us expand capacity without having to build out in our own lab by looking at a second site for our PMA test. Obviously, the economics of that will be important to us, but those are the two main channels that we're looking at to hit our growth targets.
Next question. The test appears to offer the greatest benefit in early detection of smaller lesions. If the product is cleared by the FDA, how would you like to see professional society guidelines updated to fully reflect the clinical value of HelioDx compared to ultrasound? Rick, can you take that?
Yes. Well, I think, professional societies have committees that, look at the data and evaluate them based on the strength of the evidence, And this is true both for liver societies and also for in cancer, for instance, the National Comprehensive Cancer Network. And they will assess the evidence for this. That's why we're working to get a publication out in addition to the FDA approval. And I expect, given the performance of the test, that it will enter into the recommendations. I think it may be most likely that it will come in as a preferred alternative to ultrasound rather than an absolute recommendation to replace ultrasound. But I think that's going to be determined by these committees. I'll point out that, in terms of the performance of the test for detecting earlier lesions, these are almost half of the ones in the CLIMB trial. Ultrasound missed all of them. And GALAD, which is the other multi-protein analyte test, also missed all of them. So I think the value of HelioLiver will be readily apparent.
Thank you, Rick. Next question. Can you comment on the development path for your MRD assay? Dr. Mahajan?
Yes. These are just preliminary results. We have recently started looking into the use of the methylation score, specifically for indications beyond diagnostics and looking at prognosis and MRD. And so one of the interesting things we found was that the methylation score of patients tends to decrease over time as the patients improve. And patients that progressed, the methylation score actually got worse. And so this can be used for treatment monitoring, but it can also be used in the future to measure disease levels and for making sure that the disease hasn't recurred. So increase in methylation score tends to show that something's not okay and that the cancer may have come back or that the patient progressed. And so this is something we are expanding on and working on for the next product.
Next question. What has the commercial traction been like for your first few customers? What is the most common pushback from physicians? Gary?
Yeah, I mean, so traction's again just started, and so in terms of the uptake, the expectation is very good because the physicians clearly see the efficacy of the test. That's usually where they grab on, and then after that, it really is about efficiency. It's about their ability to order the test with ease, the ability for their staff to order the test with ease, and largely the biggest pushback that we get. It's not really a pushback, it's more of an open question is what's your coverage in our area, right? They need to know that their patients aren't going to get hit with a bill later from us or from their plan in the form of an EIB. So anyway, that's kind of it. It's always around the payment when we hear pushback, and sometimes around their ability to have the test administered in their practice. But those are logistics more than anything else.
Gary, another question for you. How many salespeople will Helio need to service the market?
If we go writ large all the way to its conclusion, probably say 2 years from now, we're talking about somewhere between 30 to 50, a team about that large. So it's sales reps, it's customer experience folks that are there to manage and cultivate the clients, and then it's also other support staff. So it's somewhere between 30 and 50, depending on how we splice up the market in the U.S.
A couple questions here. I can answer these. Will Helio ever be interested in chronic disease detection or management? And is Helio developing an MCED test? We heard Dr. Rick Van Etten and Dr. Mahajan talk about our platform technologies, MESA, Eclipse, and CHOM. These are really cancer and disease and indication agnostic technologies that could be leveraged for any other type of disease or type of cancer. Helio was started with intellectual property from UC San Diego that covered 20 different cancer types. Of course, it's extremely important as a company in this field to have focus. So our first focus in beachhead was in liver cancer, where we felt we can make an impact and accurately use our resources. In order to go after an MCED test, we believe the market has to be right in terms of the reimbursement landscape, the FDA landscape, and also physician guidelines. So we don't think we're quite there yet in terms of an industry, but we do have our sights on an MCED test in the future. It's within our pipeline. We plan to develop single tissue tests like liver cancer and other cancers, and then ultimately combine them into a single assay in a modular format so that we can run thematic tests or MCED tests. All right. Next question. What is the next step for clinical validation of HelioLiver? Rick, do you want to take that, or would you like me to?
You go ahead, Justin.
Okay. So the clinical validation for HelioLiver is done, right? The CLIMB study is considered a phase 3 EDRN study with statistical significance, showing that we outperformed ultrasound, the current standard of care. The only additional data we would show for HelioLiver would be a longitudinal real-world study to show clinical utility of the test in terms of outcomes. So we are currently in discussions with various academic institutes on running these what can be called phase 4 studies, which would show cost effectiveness and mortality and clinical outcomes using our test versus the current standard of care. But that is not in the realm of clinical validation. In terms of clinical validation, we've completed that. So I think that's all the questions. So I'd like to close here by just saying, just thanking everyone for joining us. Really, at our core, we are driven by a singular mission, and that is to revolutionize cancer detection through innovation and accessibility. Early detection is not just about identifying cancer, it's about saving lives, empowering patients, and giving families more tomorrows. Our groundbreaking technology and commitment to research are paving the way for a future where cancer is caught in its earliest stages when it's most treatable. Together with the support of clinicians, researchers, and advocates like you, we can transform the standard of care and make a profound impact on global health. We look forward to your continued partnership as we move forward in this journey. Thank you.